and welcome to the Lunchtime Discovery Series. I'm your host today. My name is Miranda Dowdy, and I am coordinator of public science events here at the North Carolina Museum of Natural Sciences, which is where this edition of the program is coming from today. So the Lunchtime Discovery Series is coordinated by the Office of Environmental Education within the Department of Environmental Quality and broadcast by us here at the museum. Unfortunately, I've been having some connectivity issues, so if I drop off, just continue on like everything is fine. But before that happens, let me see if I can introduce our guests today. And it's going to be a fun show. Two of my favorite people here at the museum are today's guests. We have Dr. Julie Horvath and Dr. Marianne Barrier. Dr. Horvath is head of the Genomics and Microbiology Research Lab here at the museum, as well as the Research Associate Professor at NC Central University. And Dr. Barrier is the Assistant Head of Genomics and Microbiology Research Lab here at the museum. Hi, Julie. Hi, Marianne.
Hello.
Hi. So I am always excited to hear what you have to tell us, what you've been working on. And before I drop out, I'm going to go ahead and hand it over to you so you can tell us what you've been working on.
Sounds great. Thanks, Miranda, for the intro. All right, let me get my slides in here. I will take it away. All right. Can you guys see my slides yet? Maybe soon. All right. So today I'm going to talk about our Monkey Health Explorer project. So really learning how we can study monkey blood and help this project and also help us understand more about humans. So as Miranda said, I'm jointly appointed between the Museum of Natural Sciences and NC Central University. So we engage the public, K through 12 students, as well as undergrads and grad students at NC Central in this project. And hopefully we're going to engage you by the end. So I'm going to start out the first half, and I'm going to tell you a little bit more broadly about the project. And then Marianne is going to take it over, and she's going to tell you more specifically about how you can get involved and how you participate. All right. So if anybody's been to the Museum of Natural Sciences, you know our Genomics and Microbiology Research Lab is all glass walls. And so we're working behind the glass where you can come up to the glass and learn about what we're doing. And you can see our monkey and human ancestor skulls in the window. So this is very captivating, engaging, and you can learn more about some of the other projects. So the overarching research question lab of mine is how do host and microbial genes impact behavior and disease susceptibility. So we're going to talk mostly about the Monkey Health Project today, but we also have ongoing primate skin microbiome research, which you can learn about from past lunchtime talks or by coming to the lab and learning about some of those. So those are other projects you can get involved in. But like I said, today we want to talk about this Monkey Health Project and get you guys excited about the project like we are, and also show you how you can get involved and help us move this project forward. So these two different types of citizen science projects engage the public in different ways. So some of our microbial work actually engages the public because they come in person to the lab. They give us some of the microbial samples, microbes from their skin, and we learn how their host genetics and daily habits impact their microbes, and then how that in turn relates to their overall health. And then there are projects like the one we're going to focus on today, where we can engage the public from around the world. They don't need to come in, but you can go online to some of these online portals and connect to these projects and help us both expand and explore what we're learning about the monkeys. So one question for you guys to be a little more interactive, if you could just put in the chat. So have you participated in an online citizen science project? And if yes, tell us which platform you used. So I'm going to talk about a couple platforms, but there are other platforms that you guys may have used as well. So if you could just pop in the chat and tell us some of that, and then maybe Miranda can give us a little bit more insight into that a little later. So one of the platforms we're talking about today is the Zooniverse platform, which is people-powered research. It was started to do astronomy, and now it has a lot of different projects involved. And so you can go to this portal at Zooniverse, and you can get involved in multiple different projects, including our Monkey Health Explorer project. So anytime, every once in a while throughout the year, they feature different projects. And so when our project is featured, it's on the main page like you see here, but otherwise you can just type in the name and find it at any time. The other place you can find our project is through SciStarter. So you can go to the project finder there and you can type in Monkey Health. And so that's a great way to also connect to our project. If anybody's an educator and they want to track how much time somebody spends on the project, you can go through SciStarter and set up a portal, and then you can have your students or anybody working with you log in as well. And so then that'll actually track how many classifications they do. So it's a great way to get volunteers and get students volunteer hours. All right, so we see some stuff coming through. I see some eBird and iNaturalist, so a little bit of Zooniverse, but maybe you guys can go explore Zooniverse and SciStarter after this because you'll learn about some of these other really cool projects too. If you go to our main page on Zooniverse, this is what your main page will look like. You can also get there by just typing in monkeyhealthexplorer.org. It'll reroute you to the Zooniverse page. But this project is really focusing on, like I said, monkey health. But a lot of our collaborators study the behavior of these monkeys. The monkeys we're talking about are rhesus macaques, which I'll tell you more about later. They live on this small island of Cayo Santiago off the coast of Puerto Rico. And we're studying their behavior so we can also learn about their health and then connect that to some of our bigger projects. So we can have scientists go out and they can do focal animal follows where they actually track one monkey and watch what it's doing. Is it eating, sleeping? Is it interacting with another monkey or just sitting off by itself? And then we can also watch interactions between the monkeys. So sometimes they groom each other, sometimes they fight. And so some of that information is really useful to researchers to learn what do monkeys do and which other monkeys do they interact with. But then the other piece is that we collect some data about monkeys with their health. So we collect blood and then create these blood smear images. And so you see along the bottom, there are some pictures of blood smears. And we'll talk a little bit more about what's in one of those and get you, caught up to speed and trained on that so you guys can participate. So you don't need any skill to learn to start with, but we train you in what you need to know, and then you can go on to help with this project. All right, so rhesus macaques are really useful as biomedical models because they're our close primate relatives. So we shared a common ancestor with them about 25 million years ago. We have about 93% DNA identity. So a lot of the same genes do similar things in macaques as they do in humans. And so they're very useful from a genetic aspect as well. Like I said, we study their behavior, so they have very similar behavior to humans. So they're really useful for a lot of different aspects. They are very much used in biomedical research. So there are primate centers around the world and across the US, and they're used to study HIV, other vaccines, alcoholism, Alzheimer's disease, as well as behavioral research. So for a lot of different aspects, they're very similar to humans, and so give us a lot of insight into understanding not only monkeys and other animals, but also very much understanding humans. And if anybody has heard of the Rh factor in blood, this is a protein that's on the surface of your red blood cells. If you're Rh positive, if you're Rh negative, you don't have this protein, but it doesn't seem to be a detriment. Most people in the world are Rh positive. And I'm telling you this because one of your next questions is, do you know, just yes or no, you don't have to tell me which one, just tell me yes or no, if you know your blood Rh factor. And the reason I ask is because Rh stands for rhesus. So this Rh factor was actually identified in 1940 by Weiner and Landsteiner, who were studying rhesus macaques and identified this protein on red blood cells. And so that's how we now learned this is also relevant to humans. So again, another instance of how these macaques are really relevant to studying and learning and understanding humans. So if you could just tell me yes or no if you know your Rh factor. So it's probably not going to really be a problem unless you need a blood transfusion or unless you become pregnant. And then it's really important to know what your Rh factor is. Awesome. So like I said, I'm going to tell you a little bit more about this larger project. So some of these folks are our collaborators. So I have Patrick Jacobs, which is an NC Central intern. He's an undergrad who did a lot of the scripting work here because you can imagine we're amassing a lot of data. And so we need to go through that data and understand what the participants are helping with and then make sense of that and analyze it. Bill Studebaker in our lab has also been helping with that. And then, like I said, the second half of the talk, you'll hear from Marianne about some of the details of the project. We have Michael Platt and Mike Montague, who are at UPenn, other collaborators, and Angie Ruiz-Limbitas is at the Caribbean Primate Research Center in Puerto Rico. So she's been involved in a lot of aspects of this project and getting it going. Lauren Brent is at the University of Exeter in England, and Noah Snyder-Mackler is at Arizona State. So all these folks are really more broadly involved in this project. And like I said, we're going to talk about certain aspects of it throughout today. So I mentioned that the monkeys we're studying are these rhesus macaques, and they live on this small island called Cayo Santiago off the coast of Puerto Rico. So in the upper left is a picture of Cayo Santiago. It looks very beautiful here. And in the bottom left, you can also see some of these monkeys. So they aren't always docile and sweet-natured. Sometimes they do fight. And so here are some examples of some of these interactions. So the larger project that we're studying is really using macaques as models to study health and aging. So like I said, we're studying long-term behavior of the monkeys, then we're assessing their health with this blood smear project. We connect that with their genetics because we get blood and then we extract DNA and RNA to study some other aspects of the macaques. And then we're connecting this to some environmental factors, which I'll talk about in a minute. So if anybody knows about rhesus macaques, also known as macaca mutata, their Latin name, their extant range, their living range in the world is really mostly across parts of India and China. So these are the same species, but there are these different subpopulations in Indian origin and Chinese origin macaques. And so they're spread across a really large part of the world, but they aren't traditionally found in North America. So in 1938, Clarence Carpenter actually brought about 400 monkeys from 12 different parts of India. So he collected a lot of these monkeys and then brought them to the island so they could kind of form their own social groups. And so a lot of researchers could study their behavior. So this island isn't very big, as you saw from some of my pictures. And so all of these monkeys have kind of overpopulated the island. And early on, they would give the monkeys monkey chow from a bin. This is Clarence Carpenter and his wife now feeding the monkeys. We don't do that anymore. Now they're fed with these corrals and given, fresh food and water. And so they're given monkey chow. So the island can't support them. They do get some things from the island, but most of their food source is this monkey chow that we give them. Like I said, this island is off the coast of Puerto Rico. So anybody who's familiar with Puerto Rico, here's the mainland. San Juan is the capital city. And the place where we do a lot of the work is that small island, Cayo Santiago, off the coast near Humacao. And so this is going to be really relevant in a moment here. Currently, there are about 1,500 monkeys on this island. It definitely fluctuates with births and deaths, but they're free ranging on the island. So it's a semi-naturalistic population. They're not in cages. They can free range, free roam, and then they form their own social groups. So I said they're very social. And so they form these different clusters or social groups. The monkeys, although they look really cute and charming, they can actually carry some diseases that are harmful to humans. So if you're a tourist going around the island and you want to kayak around the island, it looks really enticing. You don't want to go on the island because some of these monkeys can carry herpes B. So this is a herpes virus similar to when we get cold sores, but their version of it is not harmful, not very harmful to them. It does cause some issues, but it's fatal to humans if humans contract it. So you want to be really careful around these animals. So we're very cautious as researchers when we go to the island. You'll notice that one monkey does have a tattoo here. So when they turn one years old, they get tattooed so we can identify them and keep track of the different animals when we're scoring their behavioral data. All right. So I mentioned that the macaques are very social, and so they form these grooming networks. And here's an example of one of those grooming networks. So the females are the circles, the males are the squares. The monkeys that are in the center are more peripheral, so they're grooming more partners and they're interacting with each other more often than some that are on the periphery. So those lines between them are connecting animals that are grooming each other. So the thicker the connection means they're doing more grooming between each other, between that pair. And so you can see that the animals in the center are more peripheral and kind of darker colors. And then those yellow males on the outside are not really part of this network, even though they're kind of in this same social group. And there are multiple social groups on the island. These have all free-formed, so researchers didn't cause these to form. The animals just kind of form these social networks. And they live in different parts of the island. So some of these social groups overlap and others are in very different parts of the island. So you can imagine that some of these animals interact in different ways and interact more than other animals on the island. And you see that the social groups are somewhere between 90 and 300 animals. So still somewhat big groups, social network-wise. And as I mentioned, there's a lot of behavioral studies ongoing on the island. So the majority of the year, every day, somebody's out there recording some of the behavioral work from some of these monkeys. And then, like I said, that connects back to some of the work that we're going to be talking about. So one topic I wanted to bring up is a paper that we recently published that was based on work from when Hurricane Maria hit the island. So in 2017, there was this massive hurricane. Hurricane Maria actually hit Cayo Santiago first. So it came across the eastern coast and then hit Puerto Rico, and it was very devastating. So not only devastated the island of Cayo Santiago, but also very much Puerto Rico. On the left here, you see a picture of Cayo Santiago before the hurricane hit. And so it looks very lush and green. You can barely tell that there are any structures on the island. After the hurricane, you see there's much less vegetation. There's not as much shade. And you can kind of see some of those, trailers and places where the scientists can do the work. A lot of those were destroyed in the hurricane and have now been rebuilt. So this is, from 2017, so it's many years ago, but it still has impacted a lot of the research ongoing here. And so we took this as an advantage to say, hey, there was this natural disaster that hit these monkeys. These monkeys are really great models to understand human health and behavior. So let's study and understand what this natural disaster did to these monkeys and see if we can learn anything more about humans and understand how that might impact humans as well. So Hurricane Maria, like I said, definitely impacted the genes and how genes were turned on and off in these monkeys. So as a monkey or any animal ages, we consider that the chronological age, are you 1 or 2 years old? But as you age, you also have different genes that turn on and off. So inside your cells are doing different things. As you get older, sometimes you need to turn different things on and off. And so your gene expression looks different. So what we did is we actually collected, we had already amassed some blood that was collected from monkeys that were before the hurricane. And then we compared that to monkeys that had blood collected after the hurricane. And we looked at the genes that were turned on and off. And what we found is that after the hurricane, there were more genes turned on that were involved in the immune system and involved in inflammation. And so if you could compare this to monkeys from before the hurricane and look at their chronological age and their biological age, and then compare that to monkeys that had been exposed to the hurricane, you could look at the chronological versus biological age. What we found is that monkeys that experienced the hurricane seem to have aged faster in a biological sense than monkeys that had not been exposed to the hurricane. So it was equivalent to about 2 years of age from this hurricane exposure, which would be equivalent to about 7 to 8 years in human years. So just imagine if we have a hurricane that hits humans and hits our coast, and just think about some of the impacts of that as well. So we're kind of learning about what's happening to the monkeys, and we can now compare that to what happens in humans. And so this is going to be really useful to see, actually, can we reverse some of these effects for some things that we do? So this is one of the really great examples of using these monkeys as a great model for understanding human health and disease and some of what we can do to translate that to humans. So as I mentioned, one of the things we want to do is now look at this monkey blood to really understand the impacts of health. So this is another way that we're looking at health, and we can do this in a lot more monkeys simultaneously. So this brings us back to this Monkey Health Explorer project. So like I said, it's a people-powered project. So we take one of these monkeys and we collect blood, and then we can create some of these blood smear images. So on the right, you see this blood smear image. You see a number of different types of cells. I've circled the RBC, or the red blood cell. So that kind of looks like a donut. It's a smaller cell. Sometimes they have an empty circle in the middle, just depending on how that cell looks for you. And then we also have platelets and white blood cells, WBCs. And so the one we're really interested in for this project is the white blood cell. And so that's where we're getting people to really help us identify and count and classify some of those. So we have about 15,000 volunteers. We really need more people because we have many, many images. We've categorized about 900 animals. So we have slides from 900 animals. We're still collecting more every year. And we've done almost about 500,000 classifications on these monkeys now. So a lot of data already, but still much more to collect. So our research goals for the project, like I said, are to examine whole blood from the macaques and use that to understand health and then connect that back to their behavior. But then we also have a training goal because we're trying to train people to identify these blood cell types by looking at these pictures. And so we go through this training protocol that Marianne's going to talk about more in just a minute. So like I said, we collect this whole blood, we take a drop of the blood, and you can smear it out on one of these microscope glass slides, and it creates that smear that you see there. And we have about 900 slides now. And then we take a microscope scanner and we scan across this slide and get a lot of different images from this one monkey. And then we can start identifying some of the cells in this. So you can see here in these yellow arrows, we've identified some of these white blood cells. And so then we get the public to help us classify which types of white blood cells they see. So here's an example of zoom in on some of these white blood cells. So there are 5 different types of white blood cells we're looking for. So neutrophils are in the upper left, and those are kind of your first responders. So if you get a bacterial or a viral infection, your body's going to make a lot of neutrophils. You can see what that neutrophil looks like inside the center of that cell. You can see these structures. And so the internal structures are what really distinguish the types of cells you have, and you can see different structures in the different cells. Lymphocytes on the right side, those are the ones that really, you have T and B lymphocytes. Those help you create antibodies, so that helps you fight off an infection. You have monocytes in the bottom left. And those kill invading cells and then kind of clean up the cell debris. And eosinophils are really interesting. So those are known for their role in allergy, but also respond if you get a parasitic infection. And then basophils we have in the middle, and you can see those look a lot different than some of the other cells. Their inner mass is a lot more punctate with a lot of dots, and they're really involved in asthma. So these are some of the white blood cells that you will encounter if you participate in the project. And like I said, Marianne's going to walk you through this. And even if you don't have any experience with this, you get trained up through the project and you get experience participating. So my final question for you is, what do you think is the most common type of white blood cell in humans? And then a corollary to that is, do you think the same one is most common in the macaques? So why don't you guys answer in the chat while we transition between my talk and Marianne. So I'm going to hand it over to Marianne while you guys start answering what you think the most common type of white blood cell is.
Thank you so much, Julie. So I was coming up with so many questions to ask you during that talk. I also started thinking of like, so when I get my blood taken, is someone doing a smear and like counting my white blood cells and stuff to give me that data?
So that's a great question. Sometimes yes, and sometimes there's an instrument we can run your blood through that actually counts them automatically by size and shape of cell. And so that's something that's more automated and faster. But there are clinicians that actually look through a smear for humans and count about 100 cells and then get a diagnosis of what the percentages of each of those types of cells.
Yeah, that's super interesting. My white blood cells are always run a little bit higher. And so my doctors think I just have a lot of white blood cells.
Interesting. Yes. I mean, there's a lot of variation from person to person, animal to animal. So I think it's really cool to study this more and learn, some of this variation and what it might mean.
Yeah. Thank you so much. All right. And I assume we're saving questions for you for the end.
Ideally, yes. So we'll let
Okay.
Marianne get
Perfect.
through hers and then we can do questions. Thanks.
All right.
Okay, so are we switched over to me now?
Looks like you're good to go, Marianne.
Okay, great. So I am going to know, you've learned all about the project and about these monkeys. I'm going to take you through actually how it works, how the project works and how you can identify the white blood cells. So first off, going back to our project, here is our Monkey Health Explorer. As Julie mentioned, if you go to monkeyhealthexplorer.org, it'll take you right to our page. It has information about the project, but you can also just jump right into this Get Started area where it gives instructions here, but you start with the beginner practice. It says beginner practice start here. And so what happens is that the first time you get to the project, it'll open up a tutorial or open up this workflow and tells you a little bit about how to look at the slides, how to look at the different cells, how to work the tools in Zooniverse, a little bit of what we call our Goldilocks test, which is telling whether the cells are, how they're spread, if they're too spread out, if they're too thick, or if they're just right. And then we have information on how to actually click on the white blood cells and identify them, and even a little video to help through. And in the beginner practice, it has images that you can try out, click on the ID, and then you can click on the next picture and it shows you the answers. So it's a good way for our beginners to get started. And then once you've done that and are ready to go, you can go back to the main page and choose one of our regular workflows. And I'm going to go to our ID white blood cells single cell test, which is something we're starting out a little new thing. Instead of big pictures, or doing small pictures that just have a single white blood cell in the center, makes it a little easier and it's more mobile friendly. So this is an example. Again, the first time you go there, it will actually pop up a tutorial that walks you through that specific workflow. But basically, the key is you just look at the center, the white blood cell in the center. You can zoom in if you want to, look really closely at the structure, and then identify what type you think it is. And then there's a field guide on the right that has lots of example pictures and other help information. So it's accessible all the time. And then once you're done, one of the cool things on Zooniverse is you can click done and get another picture, move on to the next one, or you can click done and talk. which takes you to this page, allows you to make a comment. You may be like, oh, hey, this is really cool looking, or, you know, some other comment. So it's fun because we get to interact with our volunteers. They can, you know, talk with the researchers either about a specific image, or up here on the project, if I click on the talk, there's actually a notes chat, which is about the different images. There's general chat, there's technical support, and there's talk about research. So it gives us a fun interaction with our volunteers and lets them actually talk with the researchers and learn more. And like on the chat board, we even have information for students who are helping us out as part of their volunteer hours. And so we can actually they can track their volunteer time and get credit for school or wherever they need volunteer hours. So there's some fun features. We've had some nice discussions with our volunteers through this chat, the talk option. And you might also notice this big blue banner. I even put up there that we're doing this presentation today. So maybe someone from our group will join us. But you'll notice we also have some comments here. We've had a couple of volunteers who have stepped up and offered to translate our project into other languages, which is really cool because it allows people to see the information in whatever's their most comfortable language. Right now, we have, in addition to English, we have French and Arabic. And we also have someone working on Chinese and German at the moment. So we're hoping to expand even more, but it works nicely. So when you click on the About, there's information about the project. And if you switch the language, you can see how it'll switch it to the chosen language. So it's a great addition to the project and allows us to reach more potential volunteers. But just to show you a little bit on here, this is a lot of what you learned today already, was about the project. There's some information about our team members, even a poster from a Citizen Science Conference from 2021. But on the Education tab, we also have this, which is lesson plan materials for teachers. And it's a way for bringing this project into the classroom. So I'm now going to switch over to my slides. Okay, are we good there? So, okay, what we have here is the lesson plan materials that we had an AmeriCorps volunteer, Aisha Merrick, who helped us in creating these lesson plan materials. We've got activities, worksheets, data from the project that's actually allows students to really get involved in parts of the project and learn these different aspects about blood and how that relates to health. I've also had the opportunity to present about this project in middle school classes through the SciMatch program, which connects researchers with teachers, with 7th grade teachers, and I've done that for a couple years. So I'm going to take you through a little bit of what I do when I take it to the classroom so that you can learn a little bit about how to identify these blood cells. But first, I'll tell you a little, even before we created these materials, we had some comments in our talk board from teachers, like the top comment here, as well as students in the lower two, who the teacher had already been incorporating our project into their classroom, even before we had made these materials. And we've also had students, like veterinary students or med techs, who are learning how to identify white blood cells in classes, and they're using our project as practice. So that's been a fun aspect, and especially that they've left comments about how much fun it is for them to get to use this as part of their practice. So going back to the classroom, when I bring this into a classroom after introducing the project and a little bit about the monkeys and what you've already heard, then we go into here and I say, okay, well, we have this picture here of 3 of the rhesus macaque monkeys on Cayo Santiago. So from our group. And the one in the middle looks like it might be a little tired, maybe not feeling good. And so what I do is I say, okay, well, this monkey in the middle is maybe feeling a little bad. So we're going to look at this monkey's blood and figure out what might be going on with their health by looking at their white blood cells. So just like when we go to the doctor and have our blood drawn. So one thing I like to do is we don't name the monkeys because they're not pets. They live on this island. But for today, I'd like to for you guys to give me a name. What should we call this monkey while we're working on this, talking about it? So give me some suggestions in the chat. And Miranda, if you'll just pick one to tell me, and that's what we'll go with. So, but one of the things I think with maybe this is the connection with behavior and health is, I know for me, when I'm sick, I'm typically don't, I'm kind of sluggish, maybe don't really want to talk to people, don't want to interact as much. So we may see similar kind of behaviors in the monkeys when they're not feeling well as well. So, first off, before we start looking at the blood at that monkey, we're going to learn a little bit about how we look at the blood. So as you saw earlier, for each of these animals, we take a sample of the blood and smear it across a microscope slide, stain it with some stain that allows us to see the cells more clearly. And then we can look at it under a microscope, or have the microscope images scanned so that we can see it more clearly. And so here I've zoomed in on a particular microscope image of the blood smear. And what we see here is a lot of different cells. You saw earlier that we have a couple different kinds of cells. The round discs that are all throughout. You guys have an idea of what kind of cells those might be? I'll just let you think about that for a second.
So Marianne, while folks are thinking about the cells, we only had 2 name suggestions, and I think they work well combined together. So we have Sourpuss David.
Oh. Yeah. So we'll call him Sourpuss David, or maybe I'll just call him David.
Yeah, it's a little shorter.
I feel bad, if I would also be that way if I'm sick. So, yeah. I understand. So if we're looking at David's blood here, okay, we got these cells. So the ones I'm looking here, it was ones that were pointed out before. These are the ones that are most common in our blood, and that would be our red blood cells. And in the classroom, of course, I talk a little more about what they do, the function of moving oxygen through our body. And then we have these 2 larger cells. So again, if you have any idea of what they might be, any guesses, of course, these are the ones that we're most interested in for this particular project. And these are the white blood cells, which of course are immune cells. And so they're the ones that help fight off infection. And then finally, there's the little bitty cells there. So does anyone have an idea of what the little round cells are? See if anyone can possibly give a guess. The key here, a hint, is if you're clumsy like me and get a lot of paper cuts or scrapes, these are really important to help stop the bleeding. So I don't know if we have any suggestions out there, but I'll go ahead and move on and say these are the platelets. So they're the ones that form clots to stop bleeding from happening. So these are the basic cells. Again, we're going to focus on the white blood cells. So in our classroom, the students have paper copies of these guides. This is also available as one of those field guides in the Zooniverse page. And there's example pictures of the 5 different kinds of white blood cells and a little information about how you can identify them. And so we're going to go through a few of these examples to see if you can learn how to identify some of these white blood cells. And then I'm going to test you on it and see how you do. So here are just some example images of the 5 types of white blood cells. In those same pictures, of course, you have red blood cells surrounding them. And there's even a little platelet in that top left picture. So the 5 cells again, the white blood cells, so we've got neutrophil, eosinophil, basophil, monocyte, and lymphocyte. So in the classroom, I like to go through these and give some hints a little bit about what they do, how to identify them by looking at the shapes and the sizes. And so we're going to do this. I'm going to focus first on the first 2. And I like it, look at these 2 together because they have some similarities and can get confused for each other when you're doing IDs. So the one on the left here is the neutrophil. And as you've learned, this is the one that is our first defense, fights off bacterial and fungal infections. And actually, that reminds me. So before we get started, I'm going to go back to this page. Did we have any answers for which are the most common type of white blood cell?
We do.
So what did people think?
So some people said lymphocytes. There were a couple lymphocytes, a couple said neutrophils. I think it was about split. half and half.
Okay.
So those were the two top contenders.
Okay. So in humans, the top number is actually neutrophils. Those are the ones that are the most common in humans. And so that is the one, again, because it is actually our first line of defense, we have the most of those type. So those of you who guessed neutrophils, very good. It's lymphocytes was a good guess because that is our second most common type of white blood cell. So it's not surprising that that would be one. If someone knows a little bit about blood, they might think that one was the most common. So, okay, so let's get back to figuring out how do we identify these cells? So showed you first the lymphocyte, the neutrophil, and now the eosinophil. Eosinophil on the right, it's showing you, I first show a little bit again about what they do, the different functions. So the one on the left is the neutrophils, more bacterial and fungal infection, eosinophils, more parasites, also have been shown to attack some cancer cells and part of allergic and asthma response. So how do we identify them out of the smear? Well, one, just obvious looking at them, they're much bigger than red blood cells, about 2 to 3 times as big. So they really stand out when you're looking at the spread. Both of these are about that same size. And the next thing to look for is the shape. So there's like a really dark shape inside the cell. Does anyone have any guesses in the chat of what that dark shape is? What are we looking at here? So that's often what we use to identify the different types. And you'll notice neutrophils have a really dark, like 5 blobs that kind of look like beads on a string. Whereas the eosinophil has kind of 2 big blobs, kind of looks like a pair of lungs sideways. And so that's one of the big differences in what these, how the different ones can be identified. You'll see fewer shapes in in the center of the eosinophil than you will see in a neutrophil. Neutrophils usually have 3 or more. Sometimes they have quite a lot, but eosinophils just 2 or 3 of these dark shapes. So that's one way to really identify them, particularly of, comparing to each other. Another factor to look at is the background. So the background is the cytoplasm, so basically the liquid part surrounding the, did anyone guess what was inside the cell? Do you have a guess, Miranda?
I would say microbiology is not a great area for me, which is why I love talking to you, because I'm always learning.
Well, think about when I think about when I was a kid and we had to make cell models, and there was always like a big blob in the center of the cell that was important. It contained really important material that told the cell what to do. It's the nucleus. So it holds the DNA. So the nucleus grabs the stain, it shows up more darkly so we can really see it. So you might notice that we don't see any blobs like that in the red blood cells around them. And once red blood cells are matured, they actually lose their nucleus. So that's why they don't have little circular dots, at least in humans. That's actually different in other species. That's something that we're possibly moving into in a future time, is some additional type of species blood. So the nucleus, the shape of the nucleus is important, and then the cytoplasm, that the space around it, that liquid, is another way to compare these. So neutrophils, you see it's really pale, and in the eosinophil, it's a lot darker. It kind of looks shaded. And the reason is that there's these small granules in the background of the eosinophils. I like to think of it as kind of like it has sand in it that's given it this shaded look. And initially you might think, oh, well, that's easy. I can look for that really pale pink for neutrophils. They'll always look like that. And eosinophils will always be that kind of dark pinkish purple. Unfortunately, from slide to slide, and from year to year, the stain may vary a little bit, and it may be a little brighter or darker. So instead of looking for a specific color, I like to give a little trick of look at the red blood cells that are nearby. So especially if you have the donut shaped ones that have the little light spot in the middle, the center of the donut looks about the same color as the background of a neutrophil. So if the neutrophil is about as light as the center of the donut type red blood cells, then it's more like a neutrophil. If it's darker, like the outer ring, that's more likely to be a eosinophil. So that helps the relative, like what to look for. especially if the picture happens to be really dark or really pale compared. So that's a little trick for helping identify the difference there. And then, of course, looking at how common they are, again, neutrophils are about 40 to 60% of the white blood cells in humans. These numbers are from humans. We're still working on what's the normal for rhesus macaques, and I'll show you some of that data later. But eosinophils are a lot less common, only 1 to 4% of our white blood cells. So if you have a cell, you're not sure which one it is, it's more likely to be a neutrophil. So it's good to keep that in mind if you're doing this project and trying to identify white blood cells. How common something is can help you figure out which one it's more likely to be. So now that you've learned these differences between neutrophils and eosinophils, how to look for them, we're going to do a little test. So here's an image with 4 cells labeled, and I'd like you to tell me in the chat, give me the letter of the cell that's an eosinophil. So I'm going to give you time. I want to see how you've learned to look at the cells, look at the shapes inside the cell, look at the background color. Tell me what you think. Miranda, you can participate too.
Yeah, it's helpful if I participate because then no one will feel bad for getting it wrong.
And we all have to learn at some point. I knew nothing about this level, about blood, until I started working on this project. And I've learned so much since. It's been fascinating. Yeah.
And I think I would have gotten this one right. And everyone looks like they are agreeing with me that the answer is B.
Yes, you're correct. See, you've learned. Awesome. Y'all have done a great job. Now, which one's a neutrophil? Let's see if we've got those skills. Keep going. We can get more right.
I know this one looks tough because 2 of them look similar to me.
Well,
I need a magnifying glass. Get a close look.
There can be more than one answer.
Oh, okay. Okay, let's see if
because these blood smears, the cells are just randomly spread across the slide, so you'll have different mixtures depending on which part you're looking at. So
yeah, and it looks like, yeah, people are saying A and C.
Very good. Excellent. See, you guys are doing great. Now, last question, what is D pointing to?
I need a cheat sheet. And the tiny ones are platelets, right? So it's not.
Yep, the tiny one are platelets. So what do you think D might be? Does it look like a white blood cell or not? Does it have a nucleus or not?
Doesn't look like that to me. Let's see. Folks are coming in with some answers. They're saying red blood cells.
Very good. Y'all are doing great. See, you're correct on all of it.
They're ready to jump in and start reading some slides, I think.
Exactly. So now we'll go on, and there are the next 2 ones. Now I'm not going to go into much detail because we only have so much time today. But in the classroom, I would go through more, again, comparing. Here we've got monocyte versus lymphocyte. These are 2 more red blood cells, and see how the big difference for them is their nucleus shows up as like one single shape instead of a bunch of shapes linked together. Now, actually, monocyte is actually 2 parts, but they're so close together, they typically look like one. And the big difference between them is that the monocyte is, again, 2 to 3 times the size of a red blood cell. Lymphocytes are only slightly larger. Basically, the nucleus of a lymphocyte is about the size of a red blood cell. So that one, the size can really tell you, except there's always exceptions. And sometimes there's small monocytes, and sometimes there's large lymphocytes. So again, it's always tricky. But you see that lymphocytes are the second most common. They're 20 to 40% of our white blood cells. Monocytes are about 2 to 8%, so a little less common. So you're going to see a lot more lymphocytes in our pictures rather than monocytes. And in some, depending on the stain, sometimes the monocytes have the cytoplasm actually has a little bit more of a bluish tint. That's when they switched to a different stain that gave a little more definition, which helps a lot in identifying these. And then the final one is the basophil. This is really rare. It's usually less than 1% of our white blood cells because it only responds when it's needed, basically. So it's not a lot of them floating around if you're healthy. It's only when it needs to respond to something. And it does look kind of interesting. The nucleus is really hard to see, but it's typically about a C shape or an S shape. You can kind of see this one has a C shape here, but it's covered in these dark granules that are much larger. So like compared to the eosinophil that had like what I said was like sand, this is more like pebbles. And it can vary in how it looks, but usually it's much larger blobs around it. So we do have some, but they're very rare. So in our classroom, going back to our monkey here, David, won't call him sourpuss too often, but so we've got, after teaching how to identify the cells in the classroom, we say, okay, now we're going to look at David's blood. And so we actually go through, we have these printed out images, they're laminated for the students to share and go through, and they actually go through and identify the white blood cells on each of these. So just for example, this one, how many white blood cells do you see? So go ahead and type that in, and I'll tell you a little more how we do this in the classroom. The students actually get a bunch of these, and they either work individually or in pairs or in teams, identifying all the white blood cells across all the pictures so we can get a good representation of the white blood cells from our monkey. And so, and they have a worksheet where they score it and they can total it up at the end. So for this one, I don't want to use too much of our time. So I'll go ahead and circle the white blood cells. We've got 3 white blood cells in this image. And I'll zoom in on them so you can see them a little more clearly because they're kind of small when you look at them, depending on how big your screen is. So since you've learned some about IDing, can anyone tell me if any of these are neutrophil? How about you, Miranda? Now you've learned some. Can you guess which one of these might be a neutrophil based on what you see? I'll give you all.
I'm feeling like there are at least 2 different kinds.
You are correct.
And I'm thinking that the second one might be a neutrophil.
Very good. See, you've learned. So yes, the second one is a neutrophil. And then any guess at what the outer ones are?
I'm trying to remember the order in which we've learned them.
And some of them are hard to say too if you're not familiar. Sometimes the students have a tough time saying the names of them until they, you have to practice saying these. But remember, there was
like the 2 most common ones are the eosinophil.
So the 2 most common was neutrophil and lymphocyte, but the 2 that look similar were neutrophil and eosinophil.
Okay, so they look similar to me. So are they eosinophil?
Yes, they are.
Okay.
So those are the ones that we've learned. And this is what we do in the classroom. We go through a bunch of these. I get the students to identify all the cells, all the white blood cells, and then we get back together. We total them up. We go through the answers together so they can learn more. And then in the end, we total it up and calculate the percentages and compare that to the normal range. Again, these normal ranges we're still using the human normal ranges because we're still working on gathering more data for our rhesus macaques. Which one stands out the most? So you want to notice one that seems really different from the others. I'll go ahead. I don't want to take too long. What do you think?
Yeah, I think when I did this years ago, I think when you first started it up and went on the website and took my quizzes and everything and learned. And I was like desperate to find a basophil. Like
yes.
the rarest. I was like on the hunt for a Pokemon.
Yep, exactly. In the class, I make sure to tell the students there will be no basophil, so don't try and guess that. So for this one, we ended up with a lot more eosinophils in David's blood than anything else. And so, and again, I don't have it on the screen, what the different cells do, but one of the things that eosinophils respond to are parasites. So we thought perhaps that's why our David isn't feeling good, and he's got a parasite and possibly not feeling well. And that makes sense for animals living out on an island. And So that's again how we do this through in the classroom. Many ways of incorporating the math with calculating the percentages and all that. So of course, one of the things that often comes up as a question when I brought this to classes and from other groups is, well, why don't we just use AI? Why don't we just get a computer to identify all the white blood cells? Well, we are working on that, and it's been slow. It's not that easy, but we have to. We hope that once we develop a really good training set, we'll be able to set up an AI that will be able to identify the white blood cells. But that doesn't mean the project will go away, because humans are still better at identifying unique things. And so here's an example of an image of a a blood smear where there's little dots inside these red blood cells. It's called basophilic stippling. And we had some volunteers on our project that pointed this out on some of the images from one of our animals and said, and our volunteers were actually professionally trained technologists, or I think some of them were also med students or vet students. And they said, well, if I saw this on the human blood smear, I would think that this person might have lead poisoning or some sort of heavy metal poisoning, or possibly a parasite, because you see the stippling in those situations. So we got this extra information we weren't looking for, but we actually are now learning more. And this particular monkey, we did check back with the vets, and they said, oh yes, that particular monkey had diarrhea for a while. So that is probably what happened. They probably had a parasite. So it's interesting, this project, even if we were able to get some of it more automated, we would still want our human volunteers to give us all that extra information that we couldn't easily get through the computer. So now here's kind of some summary of where we are data-wise. We still are collecting data from many different years, but 2015 is the one we have a set of 99 animals. And so we calculated the range of white blood cells of the different types in the macaques and you see them here, versus human range. You can see they actually are very similar. And the cells do look very similar. So that's how we've been able to identify them, by looking for the same structures that we see in human. So initially it looks like they do fall in similar ranges, but we're still gathering data from additional years. And the projects, we have some that are up now, that's another year, and we have some slides that have been collected recently and will be coming to us soon that we'll then add again. So that's it. For now, hopefully you'll be intrigued enough to maybe get on our project and help us out, participate, see what you can see. But we have a special thanks to all of our participants and all of our collaborators and our funders for this project. So here's our little ending slide that just has, again, all the links, our social, all of that. And if you do happen to check out our project, please leave a note in our chat that lets us know that you found us through this presentation, because it's kind of fun to know how people find us on the project. So thank you.
Thank you both so much. I really, like I said, I love hearing you talk about what you're doing any chance I get. So we had a couple of questions. So Glenn wanted to know, do all primates have the same types of white blood cells? Is that something all primates have in common?
Julie, do you want to answer that?
Yeah.
I have not looked at all primates. We have looked at multiple, and so far, yes, they all look very similar. So one of our collaborators, Noah, has gelatas. And so we have actually put some of those slides up on this monkey health page as well. And those look very similar. So if you look at other species, so Marianne mentioned this a little bit. So reptiles and amphibians look very different. Their white cells are completely different. And so it kind of depends on the species, but primarily the species that are similar in group have similar white cells and similar blood structure. So yeah, all the primates we have studied, and we probably don't have that many primates that we'll put up, but we might have a couple other primates that come into this project at some point.
And maybe you mentioned it. How long is this project continuing on? Is it just kind of like going and going?
Well, so what did we start? 2016, 2017? I don't remember when we started, 5 or 6 years ago. Maybe it was 2018. So it's been going for multiple years. And as far as we know, we plan to continue with the project. I mean, it's probably going to morph and change a little bit year to year. Because like I said, or like Marianne mentioned, there's a different pipeline now. So now you can do it with a cell phone and do more mobile. And we might change the structure if we do get AI working. So I think that was another question. Can't we just use AI to train and, machine learning and artificial intelligence to train this and get the answer right away? And even if we were able to do that, like I said, we've probably worked with 5 or 6 people who are experts in the computational field and have not gotten it to work really consistently well. I think partly because we're working on an island, and so some of the smears are different from year to year, and so there's not a lot of consistency. So it's hard to train when things look so different. So that's why humans are still the best at that. But the human piece that adds in, finding that basophilic stippling or getting vet techs and med techs to really identify things that we didn't even know to ask for. So that piece, we wouldn't be able to train AI because we don't even know what to train. And so in that sense, I think we're going to continue with the project, at least in that aspect, for a long time, because that's been really valuable and is really helping us go into new research directions. So for the short-term answer, we're going to continue this for quite some years.
All right, awesome. Thank you both so much. And we are right here with a couple minutes of time left. And I appreciate you. If you want to see the lab, it's right here on the 3rd floor of the Nature Research Center in downtown Raleigh. And they got some cool stuff in the window. You can see them in there working sometimes. You can wave hello. And again, this is a weekly program, so you can come back next week. You can see in the chat the next week, the guest is a professional photographer talking about birds. So you'll have a guest host again. And Julie, Marianne, thank you both so much. And again, both of these people are great. Come in, say hello if you get a chance.
Thank you.